The overall goal of this procedure is to utilize a modified juvenile playtest in order to view inchworming behavior, a novel motor stereotypy. This is accomplished by first socially isolating each mouse 12 hours before the testing commences with independent food and water. The second step is to set up the video camera at a 45-degree angle to the testing enclosure. Next, the isolated mice are placed in pairs within the enclosure, where their video recorded for an 11-minute duration in the dark. The final step is to analyze inchworming behavior from the video to measure duration, frequency, and latency to inchworm. Ultimately, this method allows for the analysis of motor stereopathies and can be used as an indicator for the testing of novel therapeutic interventions.
This method can help answer key questions in the autism field, such as the incidence and regulation of motor stereotypies.
Visual demonstration of this procedure is critical to learning the task because the behavioral phenotype is difficult to identify and the inchworming behavior occurs in a rapid and repetitive fashion.
Prepare the BTBR and black 6 mice for the procedure by isolating them in separate cages for 12 hours overnight prior to testing. The BTBR mice used in this study display all the main aspects of autism spectrum disorders found in mice. Black 6 mice are used as controls. The day of the experiment, place the isolated mice in the behavioral analysis room for 30 minutes.